Hello, and welcome to the official tutorial for the Fluorescence Imaging Microplastic Analysis Platform, or FIMAP, developed by me, Derek Ho. FIMAP was designed to image fluorescent microplastics under multiple excitation and filter conditions, enhancing detection and identification for researchers. In this demonstration, we'll be using Nile Red, a silver chromatic dye that fluoresces differently depending on the physical and chemical properties of the plastic it stains. Nile Red has become a globally adopted stain for microplastic detection. However, it has its limitations. False positives and inconsistent thresholding with conventional black and white imaging can reduce accuracy. To address this, we've developed an approach that integrates chemical pretreatment, fluorescence quenching, and multispectral imaging to significantly improve detection reliability. For more details, see the glowing potential of Nile Red, Ho et al. 2023. How does FIMAP work? FIMAP uses 5 excitation LEDs, 265, 310, 365, 405, and 450 nanometers, as well as 4 optical filters, green, yellow, orange, and red. These are arranged in a ring formation to ensure an even illumination, allowing the capture of 20 unique excitation filter combinations per sample. This multispectral method helps overcome metamerism, a phenomena where distinct materials appear identical under limited lighting. Let's walk through the preparation steps. For pretreatment, follow the Fenton oxidation protocol described in Dying to Know by Ho and Masura 2025 to digest organic matter and quench background fluorescence. Then use a 90mm qualitative filter paper, 2.5 microns pore size. This oversized filter can be folded and pressed into a 60mm Petri dish, reducing sample transfers and minimizing particle loss. Then, add in 15 ml of Nile Red solution. This will be prepared as a 10 micrograms per ml in a 25 to 75 acetone water mix. Place it in a 70-degree water bath for 30 minutes to enhance dye penetration. After staining, refilter to remove excess dye. Then transfer the filter paper to a flat aluminum tray for drying. FIMAP works with 3 software tools. Firstly, the Arduino IDE. This controls the LED excitation sources. Number 2, the camera control software. We could either use the Canon EOS utility or the open source DigiCam control. This tutorial focuses on the DigiCam control for accessibility, though the camera configuration applies across platforms. The third software tool is MATLAB. This allows us to perform image processing and analysis. For the camera setup, configure your DSLR with the following settings. ISO set to 100 or the lowest to reduce the noise. Shutter speed, exposure time. Adjust based on the LED brightness as indicated later. The aperture or f-stop. A wider aperture or lower f-stop number lets in more light but reduces the depth of field. So set that to its lowest as possible. So for our case, it will be 2.8. For the white balance, set it to the warmest option available, 2700K, to enhance fluorescence contrast. Depending on the camera's make and model, we can also configure other settings on the camera itself. High dynamic range, HDR, enables for clearer separation of bright and dark regions. Aspect ratio. We're able to use a 1:1 square aspect ratio to tightly frame the filter area and reduce the file size. Firstly, focus your camera using a white light source. Adjust manually or use autofocus if supported. Once focused, use the Arduino IDE to toggle LEDs. Remove the double backslash in the code to turn an LED on. Reinsert the double backslash to turn it off. Note, LED brightness varies. 265 and 310 nanometers are dimmer, whereas 365, 405, and 450 nanometers are brighter. To compensate, adjust exposure times using the following ratio: 2, 8, 1 to 1 to 1, or approximately 4 seconds to 15 seconds, 2 seconds, 2 seconds, 2 seconds. Begin imaging as follows. Insert the green filter and capture images under all 5 LED excitations. This produces image 1 to 5. Repeat the process with the yellow, orange, and red filters. Together, you will obtain 20 images, each corresponding to a specific excitation filter combination. This forms our color spectral fingerprint, the CSF. To improve detection, create an overexposed mask. For example, for condition number 12, 310 nanometers with an orange filter, increase the exposure time from 15 seconds to 30 seconds. Once you've captured your 20 images, begin analysis. Load the required files, the 20 images and the following scripts: microplastic detection _ k-means, microplastics CSF extraction, and the supporting files segmentation new and color. Set the initial parameters. Specify the mask image, example 12.jpg. Set the pixel size. Use a ruler image to determine the micron to pixel scale. And choose number of clusters, example k equals 3. Then define the minimum particle size. Here we are using 300 pixels for microplastics. Step 1. Detection. The k-means clustering will segment into 3 different clusters. Cluster 1 will be the background, cluster 2 will be the fluorescence particle region of interest, and number 3 will be the fluorescent halo. Select cluster 2, and the script will output the statistics including the coordinates, xy, area, circularity, major and minor axis lengths, orientation, and the dimensions in microns. After this, a new file, micromass selected, is generated to highlight the selected particles, and this will be used in step 2. In step 2, CSF extraction, we'll apply the mask to all the 20 images to extract the hue, saturation, value data for each of the region of interest. This yields a 20 by 3 matrix per particle, where the row represents the excitation filter conditions and the column will represent the HSV color channels. Analyzing the results. To compare the particles, we can use Mahalanobis distance, a multivariate measure of dissimilarity. A value of 0 indicates identical CSFs. A value greater than 1 indicates the particles differ by at least 1 standard deviation. For example, most microplastics differ significantly, except for expanded polystyrene and polystyrene, which is expected due to their similar chemical structures. Note on environmental samples. When analyzing environmental samples, the FIMAP code, specifically Step 1, requires a slight modification. In addition to the background, fluorescent microplastics and their associated halos, you may encounter Fenton digested biosolids, which fluoresces differently. To address this, increase the number of k-means cluster from 3 to 4 to accommodate this additional class. In this case, cluster 4 should be selected to isolate the fluorescent microplastics. Additionally, to investigate the size distribution of particle populations more thoroughly, you can modify the min object size parameter to detect smaller particles. For conservative analysis, a threshold as low as 3 conjoined particles may be appropriate. In our study, we compared the object detection across pixel thresholds of 10, 50, and 100 to assess the sensitivity to particle size variation. Here, we can see that 300 particles were detected for the smallest 10 pixel size threshold, with a minimum particle size of 24.6 microns, and 98 particles larger than 74.1 micron for the largest threshold of 100 pixels. Final thoughts. This workflow allows for an accurate, high-resolution detection and classification of microplastics using fluorescence imaging and color spectral fingerprint. With FIMAP, you move beyond basic thresholding to a data-rich, color-based method for identifying microplastics. If you have any questions or need assistance, feel free to reach out. Happy imaging!